the Nirenburg and Mattaei plus the Nirenburg and Leader experiments by Adam George. Nirenburg and Mattaei were the first to discover which specific RNA bases correlated to an amino acid. They combined poly-U, which is a chain of only uracil mRNA bases, plus a cell-free extract, which is basically the interiors of a cell containing the ribosomes, cytoplasm, etc., and finally 20 amino acids, one of which was radioactively labeled. This was repeated a total of 20 times, each with a different amino acid radioactive. Translation was then allowed to occur. If the resulting protein was radioactive, it meant that it contained the radioactive amino acids. This allowed them to discover phenylalanine correlated to UUU. They were also able to discover lysine correlated with AAA and proline correlated with CCC. The Nirenburg and Leader experiments. Nirenburg and Leader later discovered 61 codons. They took a known codon, labeled here as GUU, plus ribosomes, plus 20 amino acids, with one labeled radioactively like the experiment previous, and mixed them. They let translation occur, then poured the mixture through the filter that caught the ribosomal units. The ribosomal units also contained the amino acids and codons as they were going through translation. If the filter was then labeled radioactive, it meant that the known codon correlated with the radioactive amino acid. This was repeated for each possible codon, allowing them to discover a total of 61 out of 64 codons. These are my citations. I will post a link down below. The end. Thank you.